Thank you. Thank you. Okay, so as Kenda mentioned, welcome everyone. I'm so excited. This is a new CTN web seminar series that we're starting this year. And this is the very first session, which I'm really excited about. I've been in research for a really long time, just a little bit of a background. I started basically as a study coordinator for CTN 37 was my very first trial that I worked on in the CTN. And I kind of worked my way through all the various roles. It was, I have functioned as a research coordinator, an interventionist, a QA person, internal monitor. I've, kind of done sort of monitoring at other sites. I've managed, a large clinical trial. So this subject is very deeply embedded in my head. So I'm very excited to start this series. I feel like this is something that could benefit new researchers, folks that are new to CTN, and just people who like to make sure that they are learning things and keeping abreast of all of the changing landscape of research. But this is an introduction to clinical trials. We're just going over the basics. If you are not on mute, please make sure that you are on mute. And we are going to go ahead and get started, okay? So the agenda today is very brief, but the material is, I'm going to warn everyone ahead of time, there's a lot of text on my slides. The purpose for that is so that you could use it as a quick kind of a reference if you need it, if you're new to research and you're still trying to, get things like solidified in your memory. So we're going to go over what is clinical research in general, some terminology that is commonly used in clinical research. We'll go over the different phases of clinical trials as well as the different types of study designs. So clinical research generally, if you Google it, there's so many different kinds of definitions, but basically it's a study of safety and effectiveness of an intervention. And that intervention can be anything. It could be, well, not anything, but it could be medication, it could be devices, tools, it could be diagnostics, it could be a behavioral technique, it could be testing new technology. So it's a very broad definition.
More, are you showing slides? Because no, we don't see them.
Oh, no. That would be good. Thank you so much. I guess the share button didn't take. There we go. Okay. I'm so sorry, everyone. Whoops. Okay, so let me start over. Terminology. Here's a list of some topics, terms that will be used. If you are unfamiliar with some of these are just I tried to make them like very brief, but concise. We obviously want to know what placebo is, which is the inactive substance or medication that's provided to a control group instead of the experimental group. It does not have any active intervention. The control group is the group that is being compared to and does not have any intervention in that particular group. A cohort is a group of people with a shared characteristic. So for example, if you are the class of 02, then you're in that cohort. Target population is the group of interest that you're studying. So for example, if you are going to do a study about people with diabetes, but specifically, people with diabetes above a certain age group, that would be the target population. And the sample population is the subset that you are actually going to be including in your research study and is representative of the target population. We have other terms, including randomized controlled trial, which if you are unfamiliar with this, it's a study design that randomly assigns participants to either one arm or the other. And it could be single or double blind. An open label study is when study participants and researchers all know what the intervention is, what's being received. Double blind is when neither participant or the researchers know, and a single blind is when only the researchers know which intervention is being received by which participant. Prospective means moving forward in time, collecting data forward in time, and retrospective is referring to data in the past, so information that's already been collected or has already happened. And bias, which is an error introduced into sampling, and there's a variety of different kinds of biases, as you can see. And we're going to go over this a little bit more, but these are the kinds of things that you want to keep in mind when thinking about study design. Okay, so I'm going to do a quick little pop quiz. I want to get a sense of what the baseline knowledge is for everyone. So pardon me while I figure out how to do this polling. Well, let me put the question up first, actually. I would like you, if you don't know very much about the different phases of clinical trials, that's okay. But take a look at these questions, and there are phases 1 through 4. And if you can kind of figure out which question matches with which phase of clinical trials, that would be awesome.
Would you like us to put answers in the chat?
Yes, please. That was my next that was my next comment. Okay, I've given it a little bit of time. I'm going to go ahead and just show you the right response is D. Does the new treatment or intervention work, and what are the side effects? So let's go over. I've basically matched up the questions to each phase. There's 4 different phases of clinical trials. Actually, there's like phase 0 too, but we're going to talk about, phase 1 through 4. And the first phase is when you have a new intervention or medication. And the question you're really asking in phase 1 is it safe? And what is the right dose? And by right dose, I mean, what is the maximum dose that you can give to someone? At what point do you start observing side effects? Those kind of questions, very basic. Phase 2 kind of looks a little bit more at, does the new intervention work? Is it effective? And what are the side effects? Phase 3 is basically you're comparing it to either a standard of care or an existing treatment. And phase 4 is also more testing, but they want to look mostly at long-term sort of side effects. So phase 1, I worked several years in cancer research, and phase 1 trials can be pretty demanding. Not only are they looking at whether or not the medication is safe, they're also looking at safety. They're looking at side effects, timing, dose, as well as what's called the maximum tolerated dose. It's a very small sample size typically, and can include anywhere from 20 to 80 healthy volunteers, or in cases of like cancer research, it'd be a much smaller number, and it would be people who have the disease of interest. So, phase 1 is really important because a lot of things happen in phase 1 outside of what I just mentioned. In addition to looking at all those things, they collect a lot of samples, typically, a lot of like blood samples, urine samples. And the reason they do this is because they're doing a lot of measurements in your body of how quickly the drug works, and how much of it, or how quickly it metabolizes, and how it works generally in your body. So phase 1 can answer many, many questions in addition to just the safety and the side effects and the maximum tolerated dose. So, and they also look at the incidence as well as the change in severity of side effects that are associated with any increase in dosage. So phase 1 basically is everybody has to go through it. You have to make sure that it's safe, that it can be tolerable, and kind of be able to explain how the medication works in the body. And this obviously is for medication that I'm speaking of, but the same principles apply for if you had a new device or a new technique. You'd still have to look at safety. You just still have to look at what is the best dose and the timing, as well as any side effects that you should be looking for. Phase 2 and 3 clinical trials, I feel like most people have either heard of or worked in. And this is where you have a much larger sample size. So at least 100 people, typically can be up to 300 people. And you want to test not only the safety, but the effectiveness. And not just the maximum tolerated dose, but what is the most optimal dose? What is the least amount that you can give to someone with the maximum benefits? And they also look at different methods, a lot of times, of how to administer the medication or the intervention. So for example, I think something like ketamine. So they use ketamine in different administrations, such as nasal or intravenous, to treat certain indications such as depression. And going from an IV administration to a intranasal administration would be something that would occur in a phase 2 trial. And it is typically for people who have the disease or condition of interest. Phase 3, this is when you do the comparison of the new intervention to an existing treatment or standard of care option. And you want to look at whether or not this offers a treatment benefit to the intended population, either at the same level as the standard of care or existing medication, or is it better? This is a much larger sample size, can be up to thousands of participants, and we want to look at the population of interest. So in this phase 3, typically they receive either medication, which is the experimental group, right? Technically, or I mean, yes, experimental group, and the control group, which either gets placebo or they just get the standard of care. Randomization may occur, and that is a study design that we will discuss in a bit. But typically, new interventions have to go through phase 3 trials in order to be able to apply to the FDA for marketing approval so that this drug can become available to a much wider population. And phase 4 is asking, is a new treatment going to be okay long-term, right? They want to look at the side effects over the long term, and they also want to look at how effective it is over long-term use. Sometimes you can have a medication that works really well at first, but over a long-term use, it can become either less effective or the patient can develop some sort of tolerance to it. These are kinds of things that you want to know about new treatments. The sample size can be quite large, and it typically phase 4 trials begin after the medication or the treatment has already been approved for use in the general population for the population intended, and only occurs after you've successfully passed through stages 1, clinical trial phases 1, 2, and 3. And it can also be referred to as post-marketing surveillance. Okay, so I went through a lot of information. I know a lot of this is kind of dry, so I'm trying to break this up, and let's see, since my poll isn't quite working the way that I wanted to. I'm going to provide this question right here. So if researchers are observing the effect of a risk factor test, treatment, or other intervention without trying to change who is or is not exposed to it, what kind of study design would you think this is? Please enter your responses in the chat, and I'll give just maybe like half a minute for this. I'm really glad everyone's answering the correct answer. Okay, thank you. The correct answer is C, observational study. Yay, well done. Okay, so we have mainly observational studies, and then we have experimental studies or clinical trials or interventional studies. Those all kind of mean the same thing for the purposes of this webinar. But observational studies, kind of like it says, you observe the effect of either a risk factor, a test, a treatment, or other intervention. And the point of this is to identify and analyze patterns in the data. And experimental studies or clinical trials are aiming to test the safety and effectiveness of whatever the treatment is or intervention is. And let me go ahead and give you a few examples and definitions here. So here are different kinds of observational studies that you could potentially use. We have registries. If you don't know what a registry is, I gave some examples here of some registries. So, if you have certain types of cancer, certain health conditions, a lot of times people want to, it is still a study. You still have to consent and do all those things. But, it is more of like a tracking of all of the data and your health variables, and outcomes. A cohort study follows a group of people who share a commonality, such as exposure to a risk factor or some other intervention and how that affects their health outcomes. So it can be moving forward in time, which is prospective, or you could be looking at data that's already been collected, which is retrospective. And I mentioned the Framingham cohort study because this is one of the most, I think, like well-known cohort studies that is currently being done in the United States. It's a population-based observational cohort study. It started in the 1940s by the US Public Health Service, and it prospectively, so, when it started in the 50s, they followed those people forward in time to investigate the risk factors for cardiovascular disease. And over time, it has grown into an ongoing longitudinal study that there's just this massive database. And so that is, an example of a cohort study. A case control study compares two groups of people, those with the disease or the condition and a group of people without. So a couple of examples I can mention to you if you want to kind of conceptualize this in your mind is if a bunch of people went to a restaurant, and the foodborne illness is the thing that really jumped out at me when I look at case control studies, because you look at people who, for example, may have gotten food poisoning and you find out they all went to the same restaurant, but some people got it and some people didn't. And then you have to kind of figure out, what it is that they were exposed to that got them sick. So that's kind of my top of the head, easy example of a case control study, but there are other ways to do that as well. I'm not going to go through all of these, but I kept it on the slides, just as I said, for you to use as a reference. There are other types of observational studies here. We have a couple slides of definitions. We have proportional mortality, ecological, which is just like an epidemiological survey. We have case crossover. Cross-sectional, I'd like to point out, because this is a type of observational study that's done pretty often, especially if you've ever had to, say, do some sort of survey in school as part of your independent research credits, and you are asked to complete a survey at a single point in time. That would be an example of a cross-sectional survey. We have case control, which we just went over. And again, the retrospective and prospective cohort, you're either following a group of people ahead in time, or you're looking back at the data that's already been collected or is available. And I included this nice little table for you so that you can kind of just see very easily, why you would pick one study design over the other and its relation to time. So as you can see, most of these are retrospective, and the cohort study can be both retrospective and prospective. So this is nice for you to have if you would like to have a little cheat sheet for observational study designs. Okay, we're going to cover experimental studies, interventional, or clinical trials. So randomized controlled trials are the most common type of interventional study. I'm sure you've heard of it many, many times, and it is often considered like the gold standard of study designs. And basically you're comparing an intervention between two groups, one who gets the intervention and one who doesn't. Either they get a standard of care or they get placebo, and the group is randomly assigned to either the control or the experimental group. And that part is really important because, well, we'll discuss that in a minute. We also have pre-post study design, and that measures the outcome before and after an intervention is administered. So I will go through a few examples, but this is also something that it happens a lot, like in academia. Non-randomized trial is where you compare the control and the experimental group, but there is no randomization. And, there are a couple of benefits to using the study design. And we also have a crossover randomized control trial where participants are randomized into the experimental or the control group, and at some point are offered an opportunity to cross from one group to the other. So the randomized control trial, like I said, is widely considered the gold standard for clinical research designs. And the reason why is because first of all, there's prospective assignment. So that means you can follow people forward in time. And, that helps to establish kind of a temporality or relationship to time. And we want to, in a randomized control trial, measure like the efficacy of the new medications or the intervention. And the randomization actually helps to reduce bias. And that's one of the biggest reasons why, is to reduce bias and to be able to examine cause-effect relationships between what you're doing, your intervention or your medication, and the outcome. And another thing that's very important is that you must think about your population, your defined population, your intervention, and the outcomes that you're interested in before you collect data, before you do anything. I feel like that should be pretty obvious, but just in case anybody doesn't know, like all of these things have to be defined beforehand. Okay. A priori basically means assumptions that come during the period of a study before data collection starts, and is based on knowledge that is widely accepted as known. A pre-post study design measures the outcome before and after an intervention or medication. It can be single arm or it can be multiple arms. And, it really depends on the kind of data you're trying to collect and what outcome that you're looking at. It does have the strength of temporality, right? So if you measure something, you give an intervention, and then shortly after you give a post survey, you can kind of show over time how your intervention affected the outcome. But it doesn't account for all the other confounding variables. Any changes in your disease or condition during the study, it can't be fully attributed to the intervention, so that could be an issue as well. A non-randomized trial is comparing a control group to an experimental group, but the assignment of the group is not random. And it can be convenient, but it's just really subject to a lot of bias, because when you are not randomizing, there's a lot of, there's selection bias and there's information bias. There's a lot of things I think that can come into play that most people don't realize at the time. So randomization really helps to reduce all of those. Like I said, there are reasons for non-randomized trial. It can be easier, quicker, cheaper to administer. And depending on what you're looking at, it can be very informative. But again, the gold standard for clinical trials typically is a randomized controlled trial. And a crossover randomized controlled trial is where, if you start in the experimental group, if you start in the placebo group and later on they find that, there's just this huge effect with the intervention to a point where they, feel that it should be offered to everyone who has that condition, then the participants will have the opportunity to cross over into the other arm. This design compensates for unsuccessful randomization. But again, it is one of those things you have to carefully think about depending on what you're studying and what outcome you're looking at. Okay, time for another pop quiz for you. What study design measures the occurrence of an outcome before and again after a particular intervention is implemented? Oops, I did not mean to do that. Well, you got it right if you answer D. Pre-post study design is when you measure the occurrence of an outcome before, provide the intervention, and then take another post. Okay, I am done with my part of the presentation. Let me stop sharing. Oops, how do I stop sharing?
Go to the top of your screen and click stop share. There you go.
It's usually on my other screen. I'm using a double screen today. I'm sorry. So before we move on and I introduce you to Mark Vasquez, who will be presenting on the second part of our webinar, do we have any questions at this time? Okay, hearing none, I'm going to introduce Mark Vasquez. He is also someone who has been in research for a really long time. He was a research coordinator I don't even know, 10 years ago is when you
Yeah, about
started,
10
Mark?
years.
Yeah, at UT Southwestern. Mark and I have worked very closely together in multiple roles as well. And he's been in, I don't even know, like 6, 7 CTN trials by now.
Yeah, I think it was 6. A
6.
lot.
Which one was your first
Plenty
one?
enough. CTN 44, I believe.
44, yeah. So
Yeah.
Mark's been around a minute too, and he's great. I cannot highly recommend him enough, but I'm going to go ahead and, turn it over to you, Mark. Please feel free to let me know when you're ready for, oh wait, no, I can't because I put all your questions in.
Yeah,
You
that's
have
okay.
your-
I can just review the questions and answer them. I know all the answers, luckily.
Okay.
So-
Mark is a lot more entertaining than I am, so please enjoy.
All right, cool. Thank you. I guess let me know if you can see the screen. I should be in presenter mode. Are you able to see that?
No, I can't see any slides.
No.
Okay, then
Oh, we
let's-
can't see it.
Oh, I have to click the share button twice. Let's try this again. Okay, I think now.
Yes,
Yes.
that's better.
Okay,
Perfect.
all right, thank you. So this is the second half of the presentation, and we're did more of a zoomed out kind of approach to the clinical research. What I'll be looking at is the individual components. So and I picked essential components that I think would be helpful for the new person to research. So whether they be a research assistant or some other role, just something that they can digest. So we'll start with this pop quiz. That it won't really be a pop quiz. But the first documented occurrence of the clinical trial methodology in the Western world was found in, if anybody wants to just shout the answers, that would be, cool. Any answer?
Oyster shell.
The
It is
Bible.
It is the Bible. Thank you for that. Oyster shell was a close second, but it was the Bible. So there was a book of Daniel from the Old Testament, and he was comparing vegetarian meal with a Babylonian meal, and that's how that came. So this slide just depicts how involved research is. It's just how much there goes into it. And now we get into our agenda. So we'll discuss certain protocol components, and I kind of squeezed a few together in some places. We'll talk about human subjects protection as it involves with the participants and the researchers, informed consent form, good clinical practices and data management, and then we'll end with just some tips and hopefully helpful hints for the research assistants and research coordinators, as they're the backbone of most of our studies. So as far as protocol components goes, these are the kind of mock components that I put together. Some of them truly are, but we'll go into each one and talk a little bit about them. So the study synopsis, whenever opening a protocol, I always feel like this guy does here at the base of this huge maze, because we know that there's a lot coming. There's a lot to read, there's a lot to digest. And the synopsis is really helpful, not only for the newcomer, but it's helpful because it gives us like a snack-sized summary. I feel like that was a good way to describe it, of what the protocol is about. So when somebody approaches you and asks, hey, what do you do? What kind of a study are you on? The synopsis is a good place to refer to for a potential answer for that. Study flow is the next kind of component, I say, and this is really composed of a few other components. But what I kind of want to focus on here is, if synopsis is going to give like the why for the research, then the study flow, each of these give different parts of those W's. So like the design would provide the what, the where, and the when. It tells you exactly what's going to happen. The methods is going to provide the how. So how are we going to collect the data? Sites can then go on to create SOPs, like local SOPs, to suit their methods. And then the who is a collection of like all the humans that are involved in research. So the study population would be one, and then the study staff would be the other. So these are all important parts of of every study. And the protocol is really the first place we get to kind of get a glimpse of how we can use this information to create the study if we want to apply for the study. So, and these are kind of the sections that, that I think would be beneficial. There's a lot more to a protocol. They're all important, I'm sure, but these were, I think, to pay special attention to. Study eligibility is the next kind of big components, right? We can't have participants if we don't have inclusion and exclusion criteria. So it's the eligibility is determined by two sets of criteria, and the inclusion criteria is going to have not only like the clinical characteristic that's usually being tested for. So if you have a diabetes study, an inclusion criteria will likely be having diabetes or being a diabetic. It also covers demographic information. So you see advertisements, we're seeking 18 to 65-year-old men, that kind of thing. And all the inclusion criteria for a participant to be enrolled must be answered yes. On the opposite side, all exclusion criteria must be answered no for a participant to be enrolled. And this, the exclusion criteria helps limit, I think the most important part is it really limits increased risk to participants. Like an exclusion criteria may be for this diabetes study that there's an increased risk with the medication if you also have congestive heart failure. So they'll say that that's an exclusion criteria, and they won't be able to bring anyone into the study who has both diabetes and congestive heart failure. So it increases the likelihood of having safe participation. But the more inclusion and exclusion criteria you add, the harder it is to find participants. So the harder recruitment it may be, or the more challenging it may be. The less you have, the recruitment might be easier, but you're less refined. So maybe you're not answering that research question quite as well. So there's this balance that that research tries to do, and that's how it's done. Regulatory issues. Oops, I'm sorry, back that up. So I feel like we all have this quickly inducted into this like rite of passage of regulatory issues when it comes to CTN. There's a whole bunch of them. So I just broke these down into two categories. There's certainly loads of paperwork to pour over, but our main two like umbrella groups that we deal with as sites in the CTN are the Institution Review Board, so the IRB. And it's generally a centralized IRB, but these are people that are expecting particular submissions of paperwork, studying startup. They'll expect it during the continuing review, that's every year, and during study closeout. And they want documentation of our AEs or SAEs, protocol departures. They don't really provide they don't do any hand holding, or they don't provide checklists like the Clinical Coordinating Center does. So here at MS, we're the Clinical Coordinating Center. We're also the DSC, the Data Statistical Center. And we also require these same regulatory documents, but we're going to make sure that all the sites have them. We're going to follow up with them a lot more closely and ensure that all regulatory requirements are met everywhere. We also do audits, so monitoring. But usually regulatory affairs will come through one of these two channels, either the IRB or via MS. And lastly, for this section is participant safety. So clearly it's very important for like the study medical clinician, the doctors to understand the contraindications and what may or may not be healthy. But I feel like it's important for everyone, included right down to the RA, to at least know what to look for or know how to identify a participant that might be reporting something that could be interpreted as an adverse event. Not often does the participant report the same thing to one person as they will to another. So I think everyone having a good baseline knowledge of what safety criteria to look out for, what a participant might be complaining of, or maybe what's kind of been going on throughout the whole study. That way they can keep their eye out, because really the more people we have looking out for safety issues, the less safety issues that we have if we catch them on time. The data safety monitoring board, there at the bottom, is a collection of clinicians, medical providers that also they work with all the different sites, and they just make sure everything's being assessed in the same manner. Maybe the naming conventions are the same, and they have a final say with like serious adverse events. So when sites report those. All right, so now we're moving on to human subjects protection. And another pop quiz that I'll do myself. This president declared war on drugs and signed into law the National Research Act of, and it's out of these. So I'll give another second if anyone feels courageous enough to answer.
Reagan.
I'll say close, but not quite. All right, so we have President Nixon, actually. So I thought it was Reagan too. I was surprised, but yeah. So President Nixon not only is known for Vietnam War and like Watergate, but he also signed in the first National Research Act, and from this was born the Belmont Report 4 years post. So 1978 was the Belmont Report, and that's what we're going to look at right here. So the Belmont Report really established the ground rules for how we treat people in research. And it was a result of really identification of wrongdoings in research here in America, even that one of them is the Tuskegee research. They were studying syphilis, and they weren't consenting participants. They weren't letting them know what they had or giving them medication for it. And people died. Other people got really hurt. So anyway, this Belmont Report was brought into act, and respect for persons is the first item, and that is making sure you treat everyone with dignity, that it's a voluntary issue, so they can volunteer to or not to consent to the research. Beneficence is making sure that we do no harm and maximize the benefits while doing everything we can to minimize the danger or the harm that people might experience. Justice is making sure that those who receive the benefits, is looking at those who receive the benefits versus those who are bearing like the burden of the research. So what we'll really dive into a little further though is the informed consent form, and that comes from the respect category. So this is what we all wish we could do. At least I did when I was a research coordinator, because really we needed this consent form. We just needed the consent quiz to be done correctly without error corrections. Of course, that's not truly what we did, but that's definitely what I thought of. So informed consent being about respect, being about one of the kind of pillars of human subjects research, is a process. It's not just a 20-minute segment that we spend with the participant. It starts with the hello, and it builds credibility. We build rapport and trust. From there, we can establish the ground rules. We can set boundaries if they're not being. Well, we can just set boundaries. Sometimes it's just nice to do that right up front. And then you can maintain those communications and establish these expectations, things that you're going to continue on throughout the whole study. And as things change, as participants are going through life too, we'll be continually asking them for various different things. And this is all different levels of consent that the participant is providing, because they can take that back at any time. So we just got to keep that in mind. And that we just want them to understand as best they can what participation involves and how that continues to change as the study continues. And of course, we can't, collect any like data with the participant ID until we have full consent. So never try to collect information until we've gone through the consent process. Oh yeah, so I looked up the etymology because I just like that kind of stuff. And I thought it was really cool. So con is Latin for together, and sentire is Latin for feel. And I really think that describes what we try to do in the consent process. We try to have like a give and a take. We try to have an information exchange, and so hopefully we both feel good about it, and the participant then can have an informed decision. So here are just a few tips on conducting the informed consent. Again, we want it to be an information exchange. So it's not just a talking to the participant. We want to give them and prompt for questions that they may have, make sure we address any concerns that they have. Wording and phrasing matters. So, when you paraphrase, I think being fluent enough in that informed consent form and with the study procedures is important, that you don't misspeak and say something that's either wrong or maybe offensive or something like that. So really knowing how it's laid out helps a lot. Keeping it simple, keeping it slow. There's a lot of information and it's easy to try to burn through it really quickly, because it does take some time, and maybe you have other participants waiting, but nothing is more important than the participant you're with, and if it takes going slow for them to understand, then that's really what needs to happen. So of course, choosing a private space, a confidential space, make sure the participant feels good. And that's usually the first time we see the participant. So that's where I have, you only get one first impression. And another quiz that we get to take. So which one of these does not belong? And they're kind of long, so I don't know if anyone's going to shut this out. But I'll give it a few seconds and then I'll answer if nobody else does. Okay, so this answer is D. So study staff should sign before the participant whenever possible. That is false. These other statements are true in regards to the informed consent form. So the informed consent form is has a lot of signatures, has dates, and what we call these things, this collection of how to document, is good clinical practices, and that was the answer. And that's what we're covering next. So good clinical practices and data management. So what GCP does is it was enacted to as part of kind of the same line of thinking with the Belmont report, to protect participants, protect participation by ensuring a high level of quality for trials and creating the standard that's used internationally now for conducting research. And what GCDMP is, which is just data management kind of spiced in there, is it runs that same concept through like this lens of technology. So we have now standards that are being created for electronic case report forms and databases, one of which, if you might have heard of 21 CFR Part 11, which is like a set of requirements, specifications needed for this electronic signing of documents in lieu of paper signing. So at some point, paper signing will become obsolete, but right now you have to meet this kind of stringent list of specifications before you can do that. So most studies still have paper ICFs and use electronic databases for other things. So ALCOA is a long-running acronym that embodies a lot of the principles that we use in GCP. So the first one being attributable. I'm sorry, it was cut off at the top of my screen. And that means that we can identify the documentation. So who did it, where, and when was it? Is it dated? Legible. So we want to make sure we can read it. And that's easily is in quotations because, I mean, doctors generally don't have great handwriting, which is odd. I mean, it's not odd, but not all doctors start with bad handwriting, but they do. There was a study that I was reading, it gets worse with time. And I'm guessing that's because of the workload and whatnot. So it's not necessarily their fault that they have bad handwriting, but we want it to be legible. Contemporaneous is the data collected and then entered in real time. So the idea here is that the longer period of time that is that spans between when it's collected and when it's entered into the database, you're increasing that chance for error. So maybe if it was on a source document, you're increasing the chance that you would lose it. So we want to get that as close to each other as they can. Original, we never destroy source documents. So the original documentation should always be preserved. We want the data to be accurate. So viable data is error-free. And that's why we do a lot of our here with the CTN. We have protocol monitors, we have remote monitoring. We work with sites individually to see what kind of monitoring they're doing. So, data quality is really important, and that's something we try to stay on top of so that it doesn't build up. And being a data manager, I now have a little bit more appreciation for the efforts that goes into ensuring clean data and minimizing discrepancy. So it's pretty cool. Complete is the final criteria, and that just means that there shouldn't be any deletion or removal of data from like any source documentation. If you're going to share it with another institution, let's say, that shouldn't be just a portion of that. You need to share the whole thing, either give the whole story or don't. All right, so we will wrap it up with research assistant and research coordinator, like tips, tricks of the trade, things we picked up. So the first thing is rapport building. I think for me, this is probably the most important. It's hard to pick one thing, but it's one of the most important elements in having research with humans. The better the rapport that you build, the easier it is going to be to communicate with them. It creates like this information highway. It's not having rapport. It's so easy to lose somebody, and that rapport starts with day one, and it's built based on consistency, and it's built equally amongst all participants by like not taking favorites and things that we talked about a little earlier. Establishing an internal QA procedure. This is something that I know our site, or the sites that I was with, benefited from hugely, and that was like creating a hard rule that let's say this piece of paper does not leave this pile until somebody else comes and grabs and double checks it. And it just reduces the amount of monitoring fines. It increases the site metrics, and it gets all the team members kind of on the same page, because you're able to identify trends internally and correct them really quickly. I think it's an awesome thing to do, and it could benefit a lot more sites by really thinking about how we can introduce like QA procedures that could be not stopped or not temporary. Oops, go back. All right, so proper preparation prevents poor performance. I did say it. So this is just an ode to be prepared as much as you can. If your site is scheduled to enroll 30 participants, then make 20 participant data binders while you're waiting for your site to be endorsed or for launch. Don't wait until you enroll somebody. So thinking about things that you can do ahead of time as much as possible. When creating SOPs, really go through and see what kind of contingency plans you can come up with. Keep in mind different staff scheduling. So just be prepared and do as much kind of anticipatory action and thinking that you can. The don't do's is on this list. So a lot of errors that I've seen, I would consider them pretty common. I kind of spoke to this earlier, but don't pick favorites. So it's with our study population, it can be challenging. There's a lot of different attitudes. There's a lot of different behaviors that we either consider appropriate or inappropriate. And it's easy sometimes to like, this is my participant. And I've caught, myself doing it, other team members, because this is the person that's pleasant maybe. And I want to warn against coordinators and RAs in particular doing this because it doesn't benefit anyone. It actually could influence the participants' responses. If they're now like a friend, quote unquote, with you, right, then they'll feel ashamed self-reporting something, and now their data is tarnished. So just keeping that in mind, that everyone's equal and deserves your time in an equal proportion. Between the ears is no place for data. So this is when things are going crazy and you don't want to write something down because you don't want to create a source document out of a yellow sticky note. So you're going to try to remember this weight, let's say, or this time. Well, as you get more busy, you now forgotten that and it's gone forever. So maybe the participant never came back. But so this is I guess a call to the contemporaneous documentation. It's really important, and it'll save the site, PDs or having to deal with various queries. Like if you just take those 5 seconds or maybe 10 seconds to write down or data enter what was just collected, it's much better in the long run. And getting into that practice is just good to do as early as you can. And lastly, with CTN, again, speaking to our population that can be challenging at times. Some studies are more challenging than others. Our participants could be a little more transient in nature, and finding them after they've been missing for a while, not coming to study visits, can be a challenge. So completing as thoroughly and as exhaustively as possible the locator information form will save, hours and days worth of work in the long run. So the better you spend that 20, 30 minutes doing this locator form, the better your chances are of retaining the participant over time. So those are kind of our main tips. Maybe if Maura has any other ideas, she can pop into there. But if not, I'll hang around for any questions. I'll do my best to answer them. I do appreciate your attention, and thank you.
Bye.